All right, hello Biochem Lab. In this video we're going to talk more about our LDH lab and discuss what is going to happen during week 2 of the lab. So just as an overview and a reminder, last week we started purifying LDH from our beef heart samples. So we homogenized the muscle tissue, then we did a salting out process to try to isolate proteins with similar solubility to LDH. And we did a few centrifugation steps to collect different fractions and samples along the way. And we also ended off with some dialysis to dialyze out some of the salts from your protein sample. So this week we are going to finish purifying LDH and start preparing one sample for analysis. So to do that, we're going to do some affinity chromatography and also do a desalting spin column. And then in the rest of this lab, just to get you ready to start thinking about it, we're going to start characterizing our proteins. So we're going to do, in the next couple of weeks, an activity assay and a Bradford assay to look at how much LDH we have and how much total protein we have in each of our samples. And then in the last week, we'll do some gel electrophoresis. So for this week, week 2, we are going to start with your samples from last week. So you ended off with dialysis last week, and we swapped out the buffer for you and then placed your samples in the freezer. So before you start, make sure that sample is fully thawed. And once that's ready, we're going to then centrifuge the samples. So for just to get rid of any large solids or chunks that may have formed, we really just want to have a uniform sample to go forward with. So we're going to centrifuge in these tabletop centrifuges that are in the lab. You'll place your samples in these spaces in the rotors. Of course, make sure always with the centrifuge to balance the centrifuge, so you can choose another group to balance your tube with, just like last week. So when whoever has the lighter tube, you'll add buffer to that tube to make sure that both tubes have just about the same weight to them. And then so you'll centrifuge those samples and then measure the volume that you have. And from there, you'll remove a half ml of that sample that we'll analyze. And the rest of it, we're going to continue to purify using affinity chromatography. And that's what we'll talk about next. So let's talk a little bit more about column chromatography in general. So there are two main components to pretty much any column chromatography setup. So the first one is the stationary phase. The stationary phase is what remains in the column, and it interacts with the proteins that run through. And you also have the mobile phase. So the mobile phase is going to be that sample that you just thawed out. It's a liquid, right? And it contains some proteins, so hopefully some LDH, as well as some other proteins that have similar solubility, and they're in a buffer. And so all of those are going to travel through the column and pass through the stationary phase, which is going to interact with some of the proteins. And how do we get that mobile phase to flow through the column? There are a number of different forces depending on the particular column that you are using. So one option that is probably the simplest option is to use gravity, right? If you put liquid on top of the stationary phase, eventually, if it's packed sort of loosely enough, the mobile phase will be able to just flow through the stationary part out the other end, just using gravity. But this can be a slow process or a quick process depending on what exactly your stationary phase is made of and how tightly that it's packed. If your stationary phase is packed fairly tightly, you might want to choose one of these other two methods to help the mobile phase pass through a little bit more quickly. So the second method you might want to use is a pump. A pump is just what it sounds like. It adds extra pressure onto the mobile phase and helps to simply push it through the stationary phase. And pumps can be a little bit complicated to set up. You need more equipment. Obviously, you need a pump, you need tubing, you need connectors. There are more possibilities maybe to have leaks or errors. And so you might not want to use a pump. And for this lab, we are not going to use a pump. We're actually going to do a sort of more manual method to push the mobile phase through. And that's going to be using a syringe to push air. And the air is going to push on the buffer, and that's going to push the buffer through the stationary phase and out the other end. So let's talk a little bit more about the specific type of chromatography that we're doing for this lab, which is affinity chromatography. And this is a method that allows us to separate proteins according to their binding affinity to a binding partner. So this picture is a general diagram of what an affinity chromatography column might look like. So the column has a stationary phase that's drawn on the bottom here. So you have a solid resin matrix, and attached to the matrix is in a ligand. So it's a matrix anchored ligand. That means the ligand is anchored to the matrix, and the anchored ligand is what your protein of interest is going to interact with. So that's sort of depicted at the top of this column. So you would load your buffer containing your mixture of proteins into the top of the column. The different proteins will have different binding affinities. So our protein of interest is LDH, and it likes to bind very strongly to the component of our column that is the ligand that's anchored to the matrix, right? And so as we pass our mixture through the column, the LDH, our protein of interest, which is drawn here with these red circles, they're going to remain attached to the matrix. All of the rest of the proteins will be washed through as we add more buffer. So first we'll collect all of the unwanted proteins. While the LDH is stuck to the matrix and the ligand on the column. And then once we've collected all of our unwanted proteins, we'll wash through with a different buffer, our L-weight buffer, and that will push our protein of interest off of the matrix and out so that we can collect it. So how do we do this? What do we want our LDH to stick to? So NADH is one of the substrates that reacts with LDH. LDH likes to bind very strongly to NADH, and NADH is similar structurally to a compound called cibacron blue separose. So cibacron blue separose is going to be our ligand that's anchored to the matrix. That's part of the stationary phase of our column. And so since it resembles NADH, our LDH will stick to the cibacron blue, and all the other unwanted proteins in our sample are going to pass through the column. So for our column, here are just some general tips on how to physically do it. So the column is drawn here on the right-hand side. It's this long tube. At the bottom you have the cibacron blue separose, and the rest of it, or almost the whole rest of it, can be filled with your liquid, so a buffer. There's going to be a cap on the bottom and a cap on the top. If you want anything to be able to pass through the column, you need to remove both the caps on the top and the bottom. If you only remove the bottom, but you leave the top cap on, you'll essentially create suction inside the column, and it will be difficult for liquid to pass through. So just to be thorough, if you want liquid to pass through your column, take out both caps. If you want to stop running your column or take a break, put both caps back on. So once you're ready to use your column, there's going to be buffer that you want to push through. So to do that, you're going to use a syringe. So before we get into the more specific directions for using the syringe, one really, really, really important point: we do not want air to get into our column. If we let air get into our column material, it will mess with the flow of buffer through. It will make your life difficult. So please do not let air get into the column. So how can we make sure that happens? Well, first of all, when you're adding a liquid to your column, so throughout this process, you're going to be adding buffer, letting buffer flow through, adding more buffer. So every time you add any liquid or buffer to your column, do it gently. Don't just pour it in. If you do that, you can end up creating air bubbles and disturb the separose, the cibacron blue separose that's on the bottom of the column, and air bubbles can get trapped in that column material, and that would not help you. So when you're adding liquid, instead of pouring it in, you might want to, for example, use a dropper to add the first, say, a few mils. And then once you have a sort of space of liquid on top of your column, you can add more liquid maybe a little bit more quickly, but especially if the level of your liquid is very low, if you're close to the top of the column material, be very, very careful when you add liquid so you don't disturb the column material. So then the next tip for the actual usage of the syringe is, let's look more at this picture of the syringes that we'll be using. So the end of the syringe has a little adapter that will help it fit into the piece on the top of the column. And so make sure that when you fit the syringe onto there that you hold it all together. So the first thing you do before you put the syringe tip on to the piece of the yellow piece for the top of the column is you want to fill the syringe with air. We're not going to use the syringe to push liquid through. We're simply going to pull up the plunger, let the syringe fill with air, attach the syringe to the top of the column, and then push the air through using the plunger. But when you push the air through, make sure that you hold the syringe and the cap together with your hands and keep them all on the column. If you don't hold them together carefully, the pressure you have created with the air can make the syringe and the cap go flying. You could end up, tipping over your column or creating a disturbance with air. So please make sure you hold everything together and are very careful with everything while you're pushing the air through. And make sure when you're pushing to push gently. You don't want to push too much because you don't want to force so much air through into your column that you'll push all your buffer through and fill it with air. So when you're pushing through air, be gentle to get a sense of how fast the buffer flows through when you push your syringe. And also when you're ready to stop pushing air through, make sure you release the pressure carefully. If you don't, you could create a disturbance in your column and introduce air bubbles, which we do not want. And last, which I believe I already mentioned, but please, if you're going to take a break, just put the cap on the top and the bottom of your column if you're going to walk away from it. So that being said, let's talk a little bit more about the steps for affinity chromatography. So the first thing you'll do when you get the column, it's going to be already filled with liquid. This is the storage buffer. So the first thing you do with your column is flush the storage buffer through. And you can discard this buffer. You don't need to keep it for analysis. And so once you've done that, and the level of the liquid is just above the level of the column material, you're ready to load your sample. And you want to load your sample very slowly, not just when you're adding it in on top of the column material, but also when you're letting the sample flow through. So as your mobile phase passes through the stationary phase, you want to allow time for the LDH to interact with the Civicron blue material because the LDH needs to be able to stick. So you want to, when you're pushing the sample through with your syringe full of air, you want to do it gently and very slowly to allow time for that interaction. And at the end of this, the liquid will come out through the other end, and that's called the flow through, and you're going to collect that flow through for analysis. The flow through is going to contain most of the contaminant proteins. So these are the proteins that we collected during the salting out that had similar solubility to LDH. As you pass that sample through the column, the LDH should stick to the column material, and all those other proteins should flow through in the flow through. And once the level of the liquid there is near the top of your column material, you're ready for the next step, which is passing wash buffer through the column. So you didn't, just because of the nature of how columns work, right, you didn't let it run dry when you loaded your sample. There's going to still be some buffer containing contaminant protein sitting in the actual column stationary phase. So that's why we need wash buffer. Wash buffer is going to push the rest of that buffer through and also wash away any contaminant proteins that are still, for whatever reason, sitting on the column material that aren't bound to it. So we'll also collect the wash buffer, and that's also going to have some contaminant proteins. Should not have your LDH. So the last step is going to be to pass elution buffer through the column. The elution buffer contains NADH That's going to compete with the column material to bind to the LDH, and it's essentially going to kick the LDH off of the column and allow you to collect it in the eluent, which is what happens after the elution buffer passes through the stationary phase of the column. And so the flow through and the wash we can collect just in one big container, but for the eluent, you want to collect it in smaller 3 ml fractions. And this will allow us to, at the end, when we see what we have in each of our fractions, to isolate out which fractions might contain LDH and which fractions might not have anything in them. And that way we don't end up with a huge volume that has a little bit of protein in it. We have sort of smaller, manageable fractions, and we can pick and choose which ones we want to keep and move forward with and which ones we don't. And our eluent, just to reiterate, is going to contain your LDH, which is the protein that we're trying to collect. So finally, when you're done running your column, you are going to wash it with more buffer to clean everything out, and that buffer can be discarded. So that takes care of our first half ml sample that we collected from last week's dialysis. And now we're going to do another technique this week using a desalting column. That's a spin column. And we're going to do this to clean out some of the salts from the supernatant that we saved from our first salting out step in week 1. And we're doing this because we want to remove the ammonium sulfate from the sample, because ammonium sulfate is going to interfere with the assays that we're going to be doing in the next couple weeks of this lab. So a desalting column is similar to dialysis in that it's going to help remove salts and other small soluble material from your sample, but it's going to retain your protein and other larger molecules. And when would we want to use dialysis and when might we want to use a spin column? Dialysis is really great if you have a large sample in terms of volume, and it also is great if you have a long time, like a few days. If you have shorter time, on the order of minutes to hours, and especially if you also have a very small sample, like under a ml, a spin column is probably a better option. So that's why, since we have a small sample and we don't have a lot of time, we're gonna use this spin column to desalt our sample. So what are the components of our spin column? There's a number of different parts in this column, and please look at this diagram carefully to understand which part is which, because there's very specific instructions for how to use it, and so it's important to know the names of all of these pieces. So this diagram shows several different parts. I'll start outlining what goes in where. So let's start actually in the middle of this column. This is our sample reservoir, and that's where you're going to load your sample into. So at the bottom of the sample reservoir, there's actually a blown up insert here. There's a membrane at the bottom, right? So the membrane acts kind of like the dialysis tubing that we used last week. It has a certain molecular weight cutoff that will allow very small things to pass through, but your LDH and other larger proteins will be retained above the membrane here. Smaller salts and things can pass through during the centrifugation process. So the membrane has a support base on one side, and going back to our larger picture here, on the other side you have the retentate vial. For the beginning step, the vial is really just a cap for your reservoir so that nothing spills. We use the vial in a later step during this process. So once you load your sample in the sample reservoir and it passes through the membrane, the filtrate, the liquid that passes through is called the filtrate, and that ends up in the filtrate vial, and that's going to contain a lot of ammonium sulfate and maybe some other salts. So let's look at an actual picture of the components of our column. So here we have our sample reservoir. You're going to load in your sample through the open part at the top. It's going to sit in here. When you put it into, when you put the whole thing together and it goes in the centrifuge, the sample will pass through this membrane. This part is the container that holds the filtrate reservoir. The filtrate is what passes through the membrane when you're desalting. Here is the retentate vial, which you're going to use as a cap. And this last piece is an actual cap that you'll use at the end for your retentate vial. So here are the general steps that you're going to do. First, you're going to place your LDH sample in that sample reservoir. Then you can assemble the retentate vial. So you'll add in your LDH through when it's disassembled. This is empty, right? So you can just put the sample into the sample reservoir. Then you'll assemble this whole assembly. So you'll put the retentate vial on top of the sample reservoir, sort of like a cap, and you'll place the filtrate vial on the other side underneath the membrane. And then you'll centrifuge your desalting column until the volume is less than 1/2 ml. So you want to reduce the volume in at least half. And this will take a long time because the membrane has very, very tiny holes, and it takes a lot of force to push the liquid and the salts through the membrane. So, and then at the end of that step, the filtrate is going to contain your buffer and hopefully a lot of salt. So, well, yes, definitely a lot of salt, not just hopefully. And in the top part, in the retentate that's still contained in the sample reservoir, is going to have all of your really large molecules, all your proteins, including LDH, but probably also some other proteins. So then we repeat similar but different. So in the next segment, you'll add 1 ml of buffer to that sample reservoir that contains that retentate that your sample reservoir had this 1/2 ml in it. You're going to add 1 ml of fresh buffer to that, and then you will reassemble your whole apparatus. You'll put on the retentate vial, you'll put that on the sample reservoir, and you'll stick the filtrate vial underneath the membrane, and you'll centrifuge it again until the volume is again 1/2 of a ml or less. And this will again take a long time because it takes a long time to force the liquid through there. And this just helps us to flush out even more salts and buffer and keep your proteins in this new buffer that you just put them in that has no ammonium sulfate in it. So after that, you're ready to actually collect your sample. So in this part, we're actually going to flip things around. So in the first part, we had our sample reservoir. So this part is our sample reservoir. That was our membrane. In the first part, we wanted everything to get pushed through the membrane out into the filtrate vial. In this case, we're going to actually flip the whole thing around. Now you're going to just pull that piece out and turn it. This time, your retentate vial is going to go into the space in the filtrate vial, right? So you have a vial with a vial, and then the part of the sample reservoir with your membrane is actually up here. So in this centrifugation step, you're just pushing your sample down into this vial to collect it, because you don't want any of it to be trapped in your membrane or anywhere on the walls here. You want all your proteins to kind of be in this liquid that ends up in the vial that you're going to keep. So that's what it means by reassemble column for sample collection, is you're going to flip the reservoir around, and then you are going to centrifuge, and so that the retentate ends up in the retentate vial. And this can be a short centrifugation step, because in this step, we are not trying to force our sample through a membrane. We're just trying to collect it into a tube. So you don't need nearly as much force for that to happen, and you don't need it just takes less time. So once you've done that, then you can add buffer to your sample in the vial to bring the total volume to 1 ml. So that is what we're doing in week 2. So in summary, after this week, you are going to have separated LDH from any contaminant proteins by using the different nucleotide binding affinity from LDH. So that was with our affinity column chromatography. We will also have collected several samples for future analysis from our column, including the flow-through, the wash, and all of the fractions from the eluent. And we also used that desalting column to remove salt from our week 1 sample that we will use for further analysis, and that's from our first salting out sample from last week. So next week, we're going to finally use all those collected samples, and we're going to analyze them using an activity assay, which detects how much LDH we have, as well as a Bradford assay, which detects all the proteins that are in our sample, not just LDH. And after that, we'll do gel electrophoresis to look at the different proteins we have and sort them by size. So with that, you should be ready for this week's lab and to answer the pre-lab questions.